Welcome back to ACER Explained. In this video series, I'm going through Section 3 of the ACER Practice Test Booklet. And in this video, I'm going through questions 53 to 57, so that's Unit 17. As usual, identifying prerequisite knowledge that you might need, the reasoning skills used, as well as the bootcamp resources. So this stimulus, we are given two models for different theories on how the Golgi apparatus works. Okay, so we have two images that were given as well. And so in this image, in the first image, we're shown that, let me quickly draw it out. So we have vesicles that are sort of leading into merging into the Golgi apparatus. And then you have so this is the cisterna. This is the trans cisterna. And you have vesicles here as well that are being used to move away. So basically, we're told in the paragraph that these vesicles all contain glycoproteins, and they are meant for the passage of glycoproteins through this apparatus, right? So we have glycoproteins here, and in Model 1, they sort of move this way. And then you have a similar situation in Model 2, but in Model 2, so this is your cis. In Model 2, they don't move through the same way. You have vesicles that are sort of budding off and moving through the cisternae that way. And then at the end, you have the same vesicles forming trans. Okay, so now we're asked to find a distinction between the two models. So we have A, the glycoproteins are modified in different ways in 1 and 2. Now, we're actually not told anything about how the glycoproteins are modified in each of these models, so this can be eliminated. We have option B, glycoproteins move between cisternae in 1 but not 2. Now, this is not true because we can see that glycoproteins are being moved between cisternae. It's just in different ways. So this is eliminated. Now, C is vesicles move between cisternae in 2, but not in 1. Now, this is true. We have a unique distinction between these two, and this is that the way that the glycoprotein travels between the cisternae is through vesicles in Model 2. And in Model 1, it kind of goes through like this. So this is a good contender. Then we have D, vesicles are involved in 2, but not 1. Well, we have vesicles here and here and here and here and here. So although there aren't any vesicles involved in transporting the glycoprotein between cisternae, there still are vesicles involved. So D would be an incorrect statement, and so we can be confident that C is the correct answer. So for this question, there was no prerequisite knowledge required. However, you did need to know how to interpret diagrams and how to approach chunky text, and how to look at it systematically and not get overwhelmed by it. And we also had the process of elimination that we used in order to get to the correct answer. So the bootcamp resources that can help with this are the Reasoning Skills course, the acceleration class on diagrams and visual stimuli, and the acceleration class on chunky texts. This question is asking us about organelle Y and organelle X, and it's saying that instructions going from organelle Y to organelle X leading to protein production are in the form of what? DNA, ribosomes, transfer RNA, or messenger RNA. Now we can get a bit of an idea of what the question is asking just based off of the answer options. And if we look at our figure 1, we also are shown, we have organelle Y here, and then we have a little structure here with dots on it. And this is labeled organelle X. So for this, you do need some prerequisite knowledge about your DNA synthesis and your RNA and DNA central dogma. and protein production and how that works, and your basic cellular organelles. So here with the dots on it, you have the rough endoplasmic reticulum, and here you have the nucleus. So the instructions that go from organelle Y to organelle X for protein production, it's not going to be DNA because DNA doesn't leave the nucleus. It needs to be protected inside the nucleus, so that's incorrect. It's not ribosomes because ribosomes don't actually go from the nucleus to the rough endoplasmic reticulum. They are present on the rough endoplasmic reticulum and they do help with protein production, but they're not a set of instructions, so this is incorrect. Then we have transfer RNA, and you do need to just know that transfer RNA is that T-shaped RNA that carries the different amino acids and has the specific codon for that amino acid that allows that peptide chain to be formed during protein synthesis. So this is not a set of instructions that are going from the nucleus to the rough endoplasmic reticulum, and this is incorrect. And then we have finally the messenger RNA, and the messenger RNA are in fact the instructions that go from the nucleus to the rough endoplasmic reticulum. So you'd have a single-stranded bit of RNA, and then the codons would be matched up by the transfer RNA in order to get the correct peptides or correct amino acids in the correct order in order to form the polypeptide that the mRNA is intending to form. And so the answer is D. For this question, you did need to have a basic understanding of cell biology, including the central dogma of protein synthesis. And the reasoning skills that you could have used here to help you as well were the process of elimination. And so the resources required, that could help here are the bridging course on genetics and probability, the content in action on cells and cellular metabolism, as well as the reasoning skills course. Question 55 has some extra information here. We're kind of shown how the glycoprotein is modified from one side of the Golgi apparatus to the other. And we're told what makes up this original glycoprotein, and then what makes up the glycoprotein in its final form, or the U form, as it leaves the trans side of the Golgi apparatus. The question is asking which intermediate substance contains galactose. So now if you go back to that paragraph, you can see that the paragraph says that 2 galactose units are attached to U, okay? As well as a plethora of other things. And you realize that those other things are being added. So over here we have, for example, GLCNAC transferase, right? And over here we have mannose-sidase 2. We have another one of these, I'm just going to say GT here, and then mano 1. Then we have SA transferase. So the initial glycoprotein has 2 GLCNAC. It has 8 mannose units, and then at the end it has 4 GLCNACs. It has 3 mannose units, 2 galactose units, and it has 2 sialic acid units. And so you can see that the things that have been added at the end, there's transferases that share the same names as these final products. And so you can make the inference that these transferases are what are adding on these molecules to our glycoprotein. So now when we go back to our question, which intermediate substance contains galactose? Let's look at which substances would contain galactose. So your U is going to contain galactose because you're told that. But also, you can infer that our T is also going to contain galactose because the galactose transferase is back here and it's adding on those galactose molecules. Now, U is actually the final product, so it's not considered an intermediate substance. However, T is an intermediate substance. And either way, we're not given T and U as an option. We're only given T as an option. And so T is the correct answer here. And so here you didn't need any previous knowledge. You could infer all the information you needed to from the information that was provided. And the reasoning skills that were used here was being able to interpret diagrams and chunky text as well. And so the bootcamp resources that can help with this are the same as I mentioned for the first question in this series. So for question 56 and 57, we're given some extra information as well. We're given a graph. And so 56 is asking us, in tube 7, the most active enzyme probably does what? So looking at our x-axis, if we look at tube number 7, we see the white peak. So we have a bit of a shaded one and a darker one as well. So I'll just depict them like that. So this tube is labeled with monosidase 1 and 2, as well as GLCNAC transferase 1 and 2. So remember in our previous question, we kind of figured out the different, functions of these transferases and sidases based on what was present in our original glycoprotein versus what's present at the end, right? And so in the original glycoprotein, remember that we have 2 GLCNACs, and then we have 8 mannose units as well. And then at the end, we have 4 GLCNAC units, and we only have 3 mannose units. We have 2 galactose units and 2 sialic acid units. Right? So that's why we could assume in that last question, or we could infer, that our gal transferases were adding galactase, because at the beginning you don't have it, but at the end you do. So whichever kind of enzyme shares that name is likely the one that's going to be actually adding the thing on. So in this case, we start off with 8 mannoses, and mannosidases, and mannose. We start off with 8 mannose, and then we end up with 3 mannose. And so that means it's being removed. So the function of your mannosidase is likely that it's going to remove mannose, right? And already we figured out what transferase means from the previous question. So it's likely going to remove mannose and add our GLCNAC. So looking at our answer options here, answer option A matches this, and so that's the correct answer. So for this question, once again, you didn't need any previous knowledge, but the reasoning skills that you used were the interpreting diagrams and chunky text, as well as flowcharts, and we also were given a graph in this one as well. So the bootcamp resources that can help with this are the reasoning skills course, the acceleration class on diagrams and visual stimuli, as well as the one on chunky texts, and the one on graphs as well. So in this last question, we're just being asked which of the following is most likely, and it's referring to the tubes, all right? And so looking at the tubes, it's asking us mainly about tube number 9 and tube number 7. So in the diagram, tube number 9 doesn't really contain anything. Tube number 7 contains, let me change the color for this. It contains some cisterne looking things. and we're being asked about cisternae, so that's how I've kind of thought about cisternae. Then we have two other tubes as well. So this is 7, 9. We have 8 and 6 as well. So we have some more in here and some more in here as well. Then we also have our graph. So in our graph, we have kind of 3 peaks happening. Now this peak is at about 7. This peak is at 9. And this one is at about 6. It's a very rough diagram. We have here our GAL transferases are found, and our SA transferases are found. In this middle one, we have our monosidase 1 and 2 are found, and GLC NAC transferases 1 and 2 are found. And then here we have our phosphate transferring enzyme. Okay, so that's just a rough diagram. Now, we are trying to figure out which of the following is most likely. So, whether or not cis cisternae predominate, medial cisternae predominate in tube number 9, or trans cisternae versus medial cisternae in tube number 7. Now, in tube number 9, we don't actually have anything in our diagram, so that's why I would eliminate both A and B, right? Now, for C and D, we have tube number 7, so we have this one here. Now, it is in the middle, so you could kind of assume that the middle one would be medial, just because of these are sorted by density, and it's possible that the density of the medial cisternae could be, having the middle density. That's just if you're in a pinch and you really need to just guess and move on if you're running out of time. But let's look at the actual reasoning for this. So in tube number 9, you have your GAL transferases and your SA transferases, right? So in 6, 7, and 8, you can assume that they're going to have, because of the answer options, one of them is going to be cis, one of them is going to be medial, and one of them is going to be trans, right? So when you look at your flowchart that went from G all the way to U, you'll notice that your GAL transferases and SA transferases are the final steps that are occurring in this glycoprotein conversion. And the whole point of the Golgi apparatus is to convert that glycoprotein into a form that's ready to be transported by the vesicles from the trans cisternae, right? And so going from this cis side to the trans side, you're going from G to U. And so the enzymes that you're going to find in the trans cisternae side of things are going to be the enzymes that are closer to the end of this pathway here. And so if you go back to your flowchart, you'll find that this is GAL and SA transferases. So in tube number 9, or in tube number 8 even, which is kind of like here, in tube number 8, because 9 doesn't contain anything, remember, you're likely going to have your trans cisternae. Now, tube number 7, your monosidase 1 and 2 and your GLC-NAC transferases 1 and 2, these are the predominant enzymes here, right? And so these are the enzymes that are in the middle and the beginning aspect of this process, right? And so out of tube number 7, you only have 2 options. It's either trans or medial. And so because it's not trans, as we just established, it's going to be medial. And so your answer option here, the correct answer is D. Again, the same as before, you didn't need any previous knowledge for this question. The reasoning skills that were used are the same as I mentioned earlier, and the bootcamp resources here are the same as I mentioned earlier as well. And that brings us to the end of Unit 17. Please like, share, and subscribe for more GAMSAT help. We really appreciate your feedback. We have our Instagram here at Thank Flip GAMSAT, our podcast, Thank Flip GAMSAT, Nurses Doing GAMSAT, if you want to hear more from us. All right, I'll see you in the next one.